Thank you. What I'm going to present in this talk is multidimensional neuroanatomical subtyping of autism spectrum disorders. Autism spectrum disorder is one of the most common neurodevelopmental conditions that usually persist until adulthood, showing a couple of core deficits such as impaired social cognition, interaction, and communication dysfunction, as well as repeated behavior and restricted interest. Although the diagnosis of autism per se can be relatively well carried out in consistent manner because of the existence of these standard clinical tools, the development of an effective targeted treatment is currently quite challenged by considerable heterogeneity in multiple aspects in ASD, such as behavior, clinics, genetic etiology, and brain phenotypes. And you may know that there are many previous structural MRI studies, brain alternation in autism, but if you check the systematic review and meta-analysis, then you're going to find easily variable location and inconsistent direction of findings, which are substantially confuse the field to have a group level, a single disease mechanism in ASD. And there are many possible methodological reasons for this in terms of research design, such as mixed inclusion criteria, and also, as you see in the paper down there, it also depends on variable age ranges, and also often small sample size across the given studies that are collectively render these inconsistent findings. However, what I believe is more prevailing problem rendering this inconsistency is actually heterogeneity in brain phenotypes. However, in the majority of previous studies simply lumped individuals with ASD diagnosis into single group as if they are homogeneous disease. However, this considering individual variability is really important to address the high heterogeneity in ASD. So here what we need was a sort of unsupervised statistical learning technique which allow for letting rather the data tell us if among entire data set if there is some specific subgroup showing a similar profile, which techniques is generally called the subtyping approach. The purpose of this study was to address neuroanatomical variability in ASD. Here we propose novel multidimensional imaging-based subtyping, and secondly, to assess clinical relevance of our subtyping approach with respect to behavior symptoms and intrinsic functional connectivity networks. And finally, beyond this biological characterization, we also developed a clinically useful statistical learning framework that could predict the disease symptom severity purely based on MRI markers. So here we study autism brain imaging data exchange repository, a wide one data set that consists of more than 1,000 individuals from 17 different sites. And throughout the couple of screening procedure and MRI quality controls, at the end we had 220 ASD as well as 113 age and gender matched healthy controls, both having a T1 weighted structural MRI and resting functional MRI. First of all, from structural MRI, we extract, as you see, in 4 different morphological features: cortical thickness, surface area, geodesic distance, and intensity contrast. And these last 2 features are relatively recently proposed from the group from the UK, so I'm going to briefly explain. Geodesic distance is basically shortest path length between 2 vertices along this cortical manifold. It's not Euclidean distance, but along this cortical surface. And it's basically considered as a marker of the wiring cost of intrinsic tangential cortical connectivity. And from the study of UK group, they nicely demonstrate autistic brain shows a relatively shorter geodesic distance compared to controls. And intensity contrast is another features which highly inspired by post-mortem histological studies suggesting that autistic brain may be also affected by failure of neuronal migration during corticogenesis. So at the level of MRI, it can be reflected as a reduced tissue contrast between gray matter and white matter. So these are 4 features are previously known for being altered in autistic brain compared to controls. That's why we include these features. And we extract these features from each subject and append these feature vectors into a large feature matrix and applied fully data-driven hierarchical clustering algorithm to try to subdivide this cohort into multiple subgroups showing similar phenotypes. So let me show you the result. So it turns out that there were 3 different classes, ASD1, 2, and 3, as you see in a column. And let me quickly go through the brain phenotypes. So ASD1 shows increased cortical thickness over almost whole brain, as well as decreased intensity contrast, in other words, a tissue blurring effect. And there is a little cluster there showing increased surface area and no geodesic distance. If you check class 2, ASD2, then you're going to find this almost opposite pattern compared to class 1 because it shows cortical thinning, increased or sharper intensity contrast in visual cortex, and no change in surface area, markedly decreased geodesic distance over frontal convexity. Class 3 actually shows more of a normal range of brain variability across the first 3 features, except for geodesic distance showing increased pattern in insular areas. Our purely anatomy-driven subtyping shows also a difference in symptom severity, as you see here, based on autism diagnostic observation schedule, ADO scores. And it turns out that ASD2 and 3 shows higher symptom severity compared to ASD1, which findings are also same in subdomain scores, social interaction, and communication. Our subtyping approach was also validated independent functional subnetwork analysis using resting fMRI. Here we use NeuroThint database and put the keywords related to the mentalizing and communication, both being known for representative functional deficit in autistic brain. And then we basically constructed the functional subnetwork of mentalizing and communication, if you see it up there. And this is the result of healthy controls, and as you expected, the correlation strength is really high, showing more than 0.5 or R value. Compared to controls, however, if you check the autistic brain down there, autistic individuals shows generally decreased functional connectivity. Again, what is interesting here is, as you have seen in symptom severity, ASD2 and ASD3 shows visually higher effect size of decreased connectivity compared to ASD1. Not just stopping this biological characterization, we also built a sort of machine learning classifier to automatically predict symptom severity based on whole brain structural MRI features. And here we employed a supervised ensemble classifier, more specifically gradient boosting algorithm, so that if you see the rightmost column first, these are the selected feature among whole brain. And then using these selected cortical areas, we first train the classifier and test using unseen cases in 10-fold cross-validation with 100 iterations. So if I introduce the result, as you see, class 1, ASD1, ASD2, and ASD3, we already achieve a significant correlation between observation and prediction. And when we plot into entire data set, then we really achieve high R value and significant P value. What is interesting here is that if we proceed exactly same analysis, but without clustering information, that we actually fail to achieve as high accuracy as original framework, suggesting that our subtyping information was really necessary to predict ADO scores. So in summary, here we propose novel, non-invasive ASD subtyping approach. And our identified subtype shows gradual changes in symptom severity and anomalies in intrinsic functional connectivity. And the existence of these different subtypes hopefully reconcile so far not converging previous result. and may relate it to the presence of diverging corticogenic and maturation anomalies in disease mechanism. And finally, our prediction also clinically improved the symptom severity prediction, which support high potential utility of our divide and conquer strategy in autism diagnosis. Thank you for attention.
Yes.
Hi. So I understand you're using data set from a number of different imaging sites, right?
Yes.
I was wondering how you actually handle like differences in scanners,
Yeah.
demographic, etc.
So there were, there was that kind of different source of error effects, for example, many side effect. And here, the metric that you see is actually after statistical correction of site and age. Obviously, when we just calculate the feature without statistical correction of site, then what we found was tremendous side effect across the groups. But after this statistical correction of site, then this effect is just gone that we demonstrate. So in that sense, we correct it.
Sorry, but maybe it's because I'm not really
Sure.
well versed with statistics, but I was further wondering if, say, the sample sizes are equal across the different sites, and that whether statistically correcting for sites would prevent any one site from driving a certain effect.
So after this hierarchical clustering, we also checked the number of sample size across the site, and then they didn't really show difference patterns. They are quite evenly distributed across the three classes that we found. And then we also didn't see age effect, age difference, or IQ difference, any demographic difference in across the classes, but only for ADO scores. That's why we thought this meaningful subtyping result.
So that was a very nice talk. Did you see male-female differences in the grouping?
I'm sorry, I didn't really explain this. Our study is mainly only focused on male subjects, so we cannot really study this.
Is that what accounts for the large number of eliminated subjects?
I'm sorry?
Is that in part what accounts for the large number of eliminated subjects from the number that you started with? I noticed you had a lot.
I think original Avide data set may include female, not that I know exactly, but those three different sites that we include, there's only male subjects. So.
Hi, did you do any stability analysis for the clustering? And if you remove one of the phenotypes, would you get the same number of clusters?
But that's really great questions because we didn't do. What we did is actually bootstrapping analysis, like randomly resampling 90% of subject and recluster, and then we found consistent findings, but never really take out one of features out of 4. So maybe we have to do this and check.
And what were your, sorry, I missed things. What were your input features for the classifier?
So whole brain, cortical thickness, surface area, tissue blurring, and geodesic distance features. And first, throughout the feature selection procedure, we choose those cortical areas having relatively higher prediction power for ADO scores, and then use these cortical areas to train the classifier. So we have rigorously split feature selection, training, and testing in tenfold cross-validation.
And once you add the cluster assignment or membership numbers, then you get better classification. Is that the...
I didn't get
Sorry,
this
so
point.
once you find the cluster memberships, that improve your performance.
Yeah.
Is that correct?
Yeah.
Okay. Thank you.
Okay, we just have a time for a very briff question, and then we can save the rest for afterwards.
What were the structural features you used? Did you use diffusion, or did you just use...
Oh, maybe I was not so clear. So these are the features that I used. Cortical thickness, and the regional surface area, and geodesic distance and intensity control. These are all whole brain features, vertex-wise features. So
So your site differences that you said that you corrected for,
Yes.
they would be all different and non-linear in all these features. So just a standard correction will not work, right? Or did you correct for them separately?
Maybe that's also maybe a great suggestion. Here we assume that the whichever effect from confounding factors may be related linearly, not non-linearly. So we just when we statically correct, we use the linear model and take the residual from this linear model. But nevertheless, we never really perform non-linear model. So maybe we have to.
Thank you.
You're welcome.
Great, thank you very much.